This is the second part of the blood lecture, and we're going to talk about white blood cells. So if you haven't seen the first part, go back and do that. And so we're going to talk about different types of white blood cells and what their functions are in the body. So if you look at white blood cells, they're arranged like this. So here's the white blood cells, WBCs, or leukocytes, which is their proper name. And they're broken down into two main categories with one sort of a side category that we'll talk about. So the two main categories are granulocytes and agranulocytes. And this has to do with how they pick up stain when they're stained specifically with the kind of stain that you use to that right stain that I talked about previously. Granulocytes, when they're stained, have granules or inclusions in the cells that pick up a lot of dye, and so they look granular, like they have little spots in them. And agranulocytes do not, and so therefore they look clear because they don't pick up dye in the same way. Platelets are their whole own thing, and we'll talk about them at the end. So we're going to start with talking about granulocytes. If we go, actually, we'll just do this next one. Granulocytes are, the way I remember them is I think that granulocytes are a bunch of guys named Phil, because there are neutrophils, basophils, and eosinophils. And one of the things, so when you see things that you should be able to do on your exam are identify what a cell is based on looking at it. You should be able to tell if it's a granulocyte or an agranulocyte, and also be able to tell if it is, tell what its function is, like so why you would have extra in your body or what they do in your body. They are round in shape, and they live they're short-lived, so if you have a proliferation of them or extra of them, it's probably because you have a specific kind of infection. And they have lobed nuclei. And I always talk about them as looking like links of sausage. So sausages that in those natural casings that are hooked together. That's what I think of their nuclei as looking like. And again, they have these cytoplasmic granules that pick up stains. So they look spotted in the cytoplasm. So let's start with talking about neutrophils. Neutrophils are 3,000 to 7,000 microliters, or sorry, you should have 3,000 to 7,000 neutrophils per microliter of blood. That is the normal range. They are short-lived. They only live a short period of time, hours to days, and they divide and replicate and make more of themselves. They have a three-lobed nucleus. So here's one lobe, here's another lobe, and there's another lobe. So if you use your imagination, you can see the sausage thing I'm talking about maybe. And then they have a light cytoplasm. There are lots of granules, and they pick up the dye pretty uniformly. So you can see how it's granular, but they tend to be sort of a light purple, like lilac colored. And the purpose of neutrophils is to phagocytize bacteria. You probably remember phagocytosis from biology, but it's when one cell engulfs another cell. Bacteria are much smaller than eukaryotic cells, like white blood cells and red blood cells. And so they're very tiny, and so the neutrophil will reach out to cytoplasmic arms, if you will, and reach around and pull the bacteria inside. And sometimes fungi, but mostly bacteria. And then once those bacteria are inside the neutrophil will kill the bacteria through a process called oxidative burst. Some of the granules in the neutrophil have chemicals in them that create bleach and hydrogen peroxide. These are two things that are very very caustic, and we know of as cleaning agents. And so both of which, when they react together with the bacteria, will work to kill the bacteria. So it's a very effective way of fighting a bacterial infection. So let's return to that first slide. So don't write this down because you've already written it down. If you were to do blood work, and because you have some sort of non-specific ailment, you don't feel well, but your doctor can't figure out exactly what's wrong with you. If they find that you have, say, 14,000 neutrophils per microliter, then that would tell them that they should be starting looking for a bacterial or a fungal infection, because those are the things that excess neutrophils would indicate. And that's how that would be used clinically. All right, an eosinophil. Eosinophils, you should have 100 to 400 of them per microliter of blood. They live about 5 days, so not very long. They have 2 lobes of their nuclei, so they kind of look like a pair of lungs or a U. And then they tend to have more reddish granules than you have in the neutrophils. And again, it's harder to see on a picture because photographs have their own sort of tinges to, filters to them. But on a slide, they would look redder than as opposed to the neutrophils that look more purple. And for some reason, students always love eosinophils. I don't know, this just apparently very fun to think of them killing parasitic worms. So an eosinophil kills a parasitic worm. So you get parasitic worms, humans get parasitic worms by eating undercooked food like fish or pork. You can get them by eating undercooked pork. And then, or if you are walking with bare feet in a place where parasitic worms live, you could get them. Your animals, your pets probably have parasitic worms, or are more likely to have parasitic worms because they eat, if you have like a cat that eats wild mice, or if your dog eats things out in the woods when you're taking a walk, they can pick up parasitic worms that way. You can also get worms in your lungs, so those would be in your digestive tract. You can also get some in your lungs. That's possible as well. We live in such clean and sterile environments as modern Americans in westernized countries that this is a pretty low incidence of Americans getting parasitic worms. But in other countries where drinking water isn't very clean, or people kill their own animals and eat them, that is where you would have a higher incidence of parasitic worms. And so you find the eosinophils, though, in the tissue, in the digestive tract, and the lungs, sort of monitoring for the places where you would have, where you would get those sort of parasitic infections. So how do eosinophils kill parasitic worms? They do it by doing just what you see in this picture. I'm going to try to get rid of this. Nope, maybe not. Nope. Yeah, maybe. Whatever. Okay, so here you have this worm. It is being attacked by, this is a scanning electron micrograph, so you see it in sort of 3D-ish. And you can see the eosinophils are on top of the parasitic worm, and they sort of swarm it, and then excrete lysozyme and other digestive enzymes that break down the body of the worm, and then they digest the nutrients that they get from that and clean that up. At the same time, they would probably also cause some inflammation, which we'll talk about in our next slide. Another role of eosinophils is that they can help if you're having an allergic reaction which causes excess inflammation. Eosinophils will go and help mitigate that inflammation, particularly in allergic reactions. All right, so speaking of inflammation, we're going to talk about basophils, which are very much about inflammation. Basophils are very small, come in very small numbers, so 20 to 50 per microliter of blood. They live only a very short time, and they have, they're smaller than your eosinophils and your neutrophils, and they are characterized by these very, very dark purple granules, blue or purple. They're nucleus, well, their nucleus is purple and the granules on top of them are sort of blue, but they all kind of blend together. They look like big, dark dots. And you usually can't see that you have an S or a U-shaped nucleus in an actual histology slide because the granules cover up the shape of the nucleus anyway. So they're very dark and fairly small compared to the other granulocytes. And what basophils do is that they have histamine in them. And histamine and another chemical called heparin. And together those cause inflammation. So they will go to a site of infection or injury and release their granules. And the histamine is going to cause vasodilation, so it makes the blood vessels leaky, and that causes edema. And then the heparin is going to prevent clotting, so it's going to, generally speaking, increase, sort of add to that leakiness factor of the tissue. And so you have edema. This is a good case of, what do you call it? Athlete's foot, sorry. And so in athlete's foot, you have a fungal infection and you have a sort of raised inflammation as white blood cells, basophils will come here, cause the inflammation, and then other white blood cells will come to that, the site of that infection, and work to fight whatever is happening at that site. All right, that brings us to our agranulocytes. So granulocytes have sort of very specific categories of pathogens that they fight. Agranulocytes have not just categories, but actual specific recognized immunity. So we're going to start talking about things like vaccines, which is very timely, and so we'll see how that plays in. So agranulocytes do not have granules, as you might imagine, and they have non-lobed nuclei. So their nuclei do not look like strings of sausages. They tend to be non-lobular. All right, so we'll start with lymphocytes. So lymphocytes are more numerous. They're 1,500 to 3,000 per microliter is the normal range. And some of them are quite long-lived. There are a lot of subcategories of lymphocytes that we're going to talk. There are lots of them, and we are going to talk about a few of them that you need to know. They're small. They're just slightly bigger than a red blood cell. And so you can see them here compared for size. And they have a sort of lumpy nucleus that takes up almost the entire cell. So you just see like a little halo of cytoplasm on the outside, and they are not granular. That's how you know that they're not a basophil. So lymphocytes come in two main flavors, and then they have lots of subcategories underneath this. So you have T cells and B cells. And they are, for the most part, in lymph tissues. So in lymph nodes and there's lymph tissue in your spleen. They could be in your bone marrow. And the T cells are there. There are T cells circulating. And then there are also T cells in this lymph tissue. T cells circulating are going to go and kill other cells that are bad cells in your body. They're your cells, but they're your cells that have turned bad. So if it's a virus, what a virus does is that it infects your cells and cause your cells to make more virus. So it hijacks your body's own cellular processes to reproduce. So a virus on its own can't hurt you. It needs to infect your cells, and your cells become like a little factory for more viruses. That's why some people can't get certain viruses, because some people are immune to certain viruses naturally because those viruses can't find a way into their cells to infect their cells. So a subset of all populations will be immune for most viruses. HIV, there are some very well documented cases of people who their cells just cannot catch HIV, and their HIV can't go into their cells. And that's what we will probably find with COVID, is that there are people who the COVID virus cannot get into their cells. So what your T cells do is go around and find infected cells and kill them. So people who have a good immune response have T cells that go, and they're actually called natural killer cells, is the name of that specific type of T cell, and they go around and they poke holes in those cells, and because of osmosis, those cells rupture and die. So that's an actually very cool thing that your cells do. They do the same thing for cancerous cells. Cancerous cells are, of course, cells that are replicating uncontrollably, and the T cells, the natural killer cells, go and recognize cancer cells and do the same thing. They poke holes in them, and it's called apoptosis. Those cells die. And so they can't reproduce and make more cancer cells, and that cancer doesn't spread. So everybody has cancerous cells in their body, and your T lymphocytes take care of that on a regular basis. Mutated cells that don't look right, your body errs on the side of caution and kills those cells. And the ones that evade your immune system are the ones that are allowed to metastasize in people who get recognizable cancer. That process doesn't work properly. All right, let's move on to B lymphocytes, which is even more exciting. Let's spell antigens right. Okay, so B lymphocytes are also called B cells, and they're there to remember. They are called memory B cells, and memory B cells have seen a very specific pathogen before, a pathogen called an antigen. And so if you have antibodies for, say, I'm trying to think of something good and not controversial. If you have antibodies for the flu, let's say chickenpox, because you have all been vaccinated for chickenpox. I have not been vaccinated for chickenpox, but I had chickenpox. And those are the two different ways that you can get antibodies for chickenpox. So I got the chickenpox disease. It's a virus. I made a bunch of antibodies, and my body fought off the virus. It turns out that several hundred children per year got very sick from chickenpox and died nationally. And so when I was a kid, there was no vaccine for chickenpox, but when you were kids, there were vaccines for chickenpox. And so probably your parents got you vaccinated. And what that meant is that they injected into you a tiny dead piece of the virus. Your body, your T cell, well, stay out of the details. You have cells that recognized that antigen. Your body, sometimes when kids get vaccines, they feel a little icky for a couple days. And that's their body mounting an immune response. It takes a lot of energy. They might get a low fever. But when you do that, you have B cells that remember that. It's like a library. And they remember that antigen that they saw before. And then the next time they see that, when you come in contact with somebody who has chickenpox, then you will mount an immune response, and you will not get chickenpox, or you will not get chickenpox as badly as you would have. My sister had a very terrible case of chickenpox. She had a high fever, and she had lesions all over her body, and she has scars from it. It really, most kids survived it, and so it was not much of anything. But it wasn't like life-threatening for most kids, and it wasn't thought of as being life-threatening. But it was for a small subset of the population. So it essentially means that rather than having to ramp up a whole immune response where you get sick for a long time, your body fights it off right away, and you don't get very sick. And they do that because your memory B cells can make these cells called plasma cells, and those plasma cells will make antibodies. Antibodies can bind to bacteria or viruses and immobilize them. So here's a bacteria in this case. And the B cell makes very specific antibodies that bind to the outside of that, and then whatever the foreign antigen or the pathogen is. And then macrophages, which is another type of immune cell, which we've talked about when we talked about skin. And remember that they're literally macro big phages, eaters, big eaters. And neutrophils, if it's bacteria, will come and they consume the whole thing, both the bacteria and the antibodies. And they are drawn to the antibodies. They hone in on that. And so that's the way the vaccines work. So hopefully someday soon, we will all be able to get a little piece of COVID that is dead or somehow genetically engineered to be inactivated, and we will all be able to get little injections, and so we will have memory B cells, and enough of us will be able to, when we come in contact with it, when we come in contact with someone who is infected, you will make antibodies and you won't actually get sick from it. That's the idea. All right. Let's move on to our other friends, our other agranulate set friends called monocytes. Monocytes, there are about 100 to 700 normally, if you're healthy, per microliter of blood. And they can live for months on end, so they are long-lived for white blood cells. And they have this big fat U-shaped nucleus, and they're pretty big. And they have sort of pale blue, non-granular cytoplasm. And what they do is that they turn into macrophages that we were just talking about. So they're called monocytes when they're in your bloodstream. And then if there's inflammation or infection, they leave. They're very, this is the macrophage here, and it's eating these little cocci bacteria. But they are able to sort of squish out through blood vessels and enter tissue. So they exist in blood as monocytes and in tissue as macrophages. And they're the same cells, they just have different names because, anatomy. And they are going to, as macrophages, they phagocytize bacteria and antibodies, and they break them down. And they also, if these are novel bacteria, they will take them to T cells and B cells, and so you can mount a big immune response and start making antibodies if you're not already doing that. All right, and so the other thing that macrophages do is that they recycle old red blood cells in your liver. So your liver is the place where red blood cells go to be consumed, and they help break those down as well. All right, so back when we looked at this little diagram, where did it go? Here, nope, here, nope, here. Okay, we said that platelets are their own thing out here. And so it's time to talk about platelets that are neither, they're not really granulocytes and they're not really agranulocytes. They are, so if you're looking at this picture, you have red blood cells, you have white blood cells, and then these little things that, students always say that this looks like a salad. These little sort of chartreuse colored guys are the platelets. And you have a ton of them. You should have 150,000 to 400,000 per microliter of blood. And when we'll go back and look at a picture and we'll see all the little platelets. Once you see them, you're like, oh yeah, they're everywhere. They're itty bitty, and they aren't really granulocytes or agranulocytes. They don't have nuclei. They're little shards of a big cell called a megakaryocyte. And they are there to cause clotting to happen. All right, so here's your megakaryocyte, and it is shedding off. These little guys are platelets, and these are lymphocytes. This is in bone marrow. And so they just shed them off consistently, and they, as they do that, they make platelets. All right, and I think we're going to wait and do hemostasis and blood clotting at the end of the next lecture.